Shall I start the class, sir? Good afternoon, class. Welcome to Dr. B.R. Ambedkar Open University. My name is Dr. I. Shantipriya, and I'm here to take class on toxicology of insecticides. Yesterday, we have learned about the formulations and classifications of insecticides. I just want to briefly go through the summary of what we have learned, like routes of entry and the mode of action they do, and the chemical structure, and the mode of action according to IIAC. So today, the next chapter is evaluation of insecticide toxicity. So, yesterday we have learned about their chemical structure, their mode of action, their entry, what do they do and all. And now, today we are going to learn the evaluation of the activity of the insecticide and the toxicity as well. So, coming to the objective, am I audible? Can anyone kindly confirm? I hope I'm audible. I'm starting the class now. The unit 2, that is evaluation of insecticide activity. Coming to the objective, today we will learn the evaluation of insecticides, the parameters and the procedures for conducting their toxicological investigations, particularly on animals and insects, and method of estimating the probit values for the statistical presentation of the results. Coming to the introduction, toxicology has been considered a science of poisons affecting human life and therefore as a branch of medical science. Du Bois and Gehling in 1959 defined toxicology as the branch of medical science that deals with the nature, properties, effects, and the detection of poisons, therefore the science of poisons. Pesticides are the largest group of poisonous substances that are used all over the world. The necessity to use insecticides is to enhance food production as well as human health, force the entomologists to study the properties of the insecticides. Insecticide toxicology includes studies to determine tolerance levels of the pesticides in man and is concerned with establishing a logical basis for selective toxicity in order to control insects without affecting other forms of life. Coming to the evaluation of toxicity, toxicity of any compound is related to the dose that has been taken. So in order to be a compound to be toxic or in order to be a compound to be a medicine, what determines? Particularly the dose depends, right? So how much amount of dose you take will determine whether it's going to act good upon you or whether it is going to create ill effects on you. Highly toxic substances cause severe symptoms of poisoning with small doses as well. So if they're highly toxic, even the smaller doses are enough to create poison. And the toxicity of insecticide to an organism is generally expressed as lethal dose 50. So lethal dose 50 indicates the toxicity of insecticide to an organism, like what it causes to an organism. So what is this lethal dose 50? So this is the amount of material or the pesticide or any drug given all at once, which causes the death of 50% or one half of the group of the test population or animals. And LD50 is the lethal dose of insecticide that kills 50% of sample populations in a given time period. As we can see the image here, it is the starting population, the experimental group, the starting population, which is like 100% here. When you expose 100 mg per kg of body weight, the exposure, it is not lethal and it is below the threshold. So there is no effect of the pesticide. But after giving 200 mg per kg of body weight exposure, definitely there is the threshold level has been increased or the threshold level has been crossed, which leads to some effect in the individuals. So here, the poisonous effect can be seen in few individuals. Later, increasing the dose to 300 mg per kg exposure has led to the 50% of the population decrease. This is the lethal dose, which causes 50% of the insecticides to die. So that is what we call the lethal dose 50. The LD50 is usually expressed in milligrams per kg of body weight of the animal. Under certain conditions, the term micrograms per insect may be used when the chemical is applied topically to the insect. The LD50 is one way to measure the short-term poisoning potential, that is acute toxicity, which is an immediate toxicity of a material. Toxicologists can use many kinds of animals, but most often testing is done with rats and mice as they really mimic the human. The smaller the LD50, the more toxic the compound. Yes, the smaller the LD50 indicates that the most poisonous the pesticide is, causing the ill effects on the humans, which leads to 50% of the population decrease. An example of that is a pesticide with an LD50 of 5 mg per kg is 100 times more toxic than a pesticide with an LD50 of 500 mg per kg. Even though the concentration of the pesticide has increased, yet it depends on the severity of the pesticides, how much it is creating the 50% of the population to be affected, right? So that is the LD50. Coming to LC50, this is also one of the terms used to determine the toxicity, that is called lethal concentration. And LC50 of the chemical in external medium required to kill 50% of the sample population within a given time period. It does not occur immediately, but within a given time period, whatever the concentration causes, 50% of the sample population dead, that is called LC50. And this is expressed as a percent of active ingredient of the chemical in the medium or as parts per million. So 1 ppm is 1 mg per liter. That is the millionth of it. LC50 value is used when the exact dose given to the insect cannot be determined. So the major difference between LD and LC is that in LD, you know how much you're giving, the dose that you're giving to that particular population, which causes the 50% of the population death. But in LC50, you do not know how much dose causes 50% of the population death. But in that, you have to so there is a higher chance of getting misled with these two terms. So the terms lethal dose and lethal concentration should not be used interchangeably when presenting the results. And LD is defined as an exact amount of insecticide applied directly to an organism. Example, units of insecticides mass applied per insect, or insecticide mass applied per average mass of insecticide tested. And lethal concentrations, like that is LC50. So here in this graph, you can see the lethal uh, dose concentration, which I have shown earlier, the lethal dose concentration where the population gets killed 50%. And here in this curve, you can see the LC50, the concentration of the toxin, which causes 50%. So here, this is the concentration of the toxin, which caused the 50% of the population to be dead, and that is called the lethal concentration, which is expressed in ppm. Right? For lethal concentration, insects are exposed to or allowed to interact with some amount of insecticide per period of time, but how much they actually make contact with or ingest is unknown. Lethal time. Again, here we have lethal time is a term that is particularly used in toxicology, similar to LC50, but instead of referring to the lethal concentration, it tells us the lethal time it is needed to kill 50% of a test population when exposed to specific dose or concentration of an insecticide. So here we are not going to check about the dose or the concentration, but in lethal time, we will say how much time it has taken to kill 50% of the test population. That is known as lethal time. This test is used when the number of individuals is limited and is often employed in field tests where it is impossible to collect sufficient number of insects for testing. In some instances, the rate of knockdown rather than kill is measured as a criteria for toxicity. In such cases, KD50, that is knockdown dose, or the KT50, that is knockdown time 50, is used. And in some other instance as well, killing or knockdown dose cannot constitute the desired criteria. For example, in testing insect growth regulators or chemistry lens, the objective is not to kill the organism, but rather to affect their developmental stages and egg viability. In such cases, effective dose or effective concentration is used to describe the results of such test, because here the organism is not going to be killed, so it is not lethal, but we are just going to check that how much dose is effective or how much concentration is effective to attain what that particular stage which we wanted to see. Any value of LD can be determined from the described analysis like LD1 to LD99, but describe the importance of LD50 is the choice of 50% lethality is used as a benchmark in order to avoid the potential for ambiguity of making measurements in the extreme and reduce the amount of testing requirements. However, whenever it is required in specific purposes, LD1 to LD99 is also being used for special studies as well. But mostly and generally, most of the studies consider LD50 in order to avoid ambiguity in the results and to get maximum information and maximum accuracy. So coming to the toxicological evaluation in higher animals, Evaluation of toxicity in higher animals differ from that of insects, obviously. And it is as follows. The number of available animals for testing is usually limited in higher animals, while the process for determining LD50 is identical in both the cases. The greater emphasis is placed on qualitative rather than quantitative aspects of poisoning in case of higher animals. In higher animals, the overriding concern is the evaluation of safety for man, while the toxicological studies on insects are conducted basically with an intention to evaluate the efficacy of insecticides against insects. Chronic effects of insecticides on insects are usually of not as much importance as in case of animals. Hence, toxicological testing on higher animals includes evaluation of short-term and long-term exposure. Toxicity can either be acute, that is for short-term with high dose, or chronic, that is for long-term with slow or smaller doses. A substance with a low toxicity generally requires large doses to produce mild symptoms because it is not having much toxicity, so you have to give more number of doses to produce that mild symptoms. Even common substances like coffee and salt will become poison if large amounts are consumed or if they are consumed above the recommended dietary allowance or above the recommended dietary intake. So toxicity can either be acute or chronic. In the sense, it can be immediate or it can be prolonged. Acute toxicity. Coming to the acute toxicity is the ability of a substance to cause harmful effects which develop rapidly following exposure. That is within a few hours, you can see the harmful effects, or within a day or 2 or 3, you can see that. So it is like immediate effect. But when it is different for chronic toxicity, where the ability of a substance to cause adverse health effects resulting from long-term exposure to a substance. I have included a picture for better understanding here. When a person or any higher animal comes in contact with the exposure of the pesticide, in case of acute toxicity, what happens is that the result or the ill effects are immediately seen. However, in the case of chronic toxicity, it causes adverse health effects resulting for long-term exposure to the substance. And the trouble is the chronic toxicity is much harder to recognize, but it is still harmful. Here is a table showing the oral LD50 values for some common substances studied in rats. So LD50, we have already read like the lethal dose that is lethal to 50% of the population. And here the substances like acetaminophen, that is paracetamol. Here, studies show that 1945 mg per kg of body weight in rats is found to be LD50. Aspirin is 2000 mg per kg. Caffeine, if taken orally, 190 mg per kg of the body weight in rats is found to be poisonous for unshowed LD50. Ethanol, 7060, nicotine 50, sodium chloride 3000 mg per kg body weight, cyanide is 5 to 10 mg per kg body weight, acetone is 5800 mg per kg body weight, lead, formaldehyde, methanol, hydrogen peroxide, chloroform, and ammonia can be seen here. The oral LD50 values are estimates based on rat studies and can vary depending on factors such as age, sex, and health of the animals used in the test. After that, we'll come into pesticide and animal interactions. So animals, beside pesticides, also react to the chemicals. Not only pesticides, but there are also other chemicals that are toxic. And animals, they tend to react with other chemicals as well. And the genetic, physiological, anatomical, and biochemical variability among and within the animal species dictate the effects of the chemical or the consequences of the chemicals. Pesticides that is highly toxic to rats, they may not be toxic to dogs or humans, while another may be toxic to rats and mice, but not to fish or birds. So it basically what I want to say here is that pesticides, they may be toxic to one species and may not be toxic to the other species. However, in the similar species also, it depends on the person's sensitivity to that particular pesticides or chemical, because each person responds differently to each pesticides, and also the dosage also matters there, because some may be hypersensitive, however some may be over resistant to particular pesticides or chemicals. Again, pesticides cannot be categorized as safe or dangerous to humans merely because they are classified as a substance that kills pests. Each active ingredient has its own unique chemical structure and toxicological characteristics. More resemblance to chemical structure not necessarily produce similar effects with regard to toxic effects. So these points has to be considered here. Effect of the pesticide on the animal. So according to in vitro and in vivo studies that are used to predict and explain the toxicity of a pesticide, effects on animals are determined by the chemical structure of the pesticide, its mechanism of action, and the fate of the chemical within the animal. The effects of a pesticide may not be similar to different animals. The effects are not only species specific, but also individual specific, like how we discussed earlier. This is an image like how pesticide enters into different, how we come into contact with the pesticide. Here, farmers, the one who is the applicator, the farmer, the humans, animals, plants, beneficial species, aquatic animals, soil microbes, higher animals. So everybody are getting affected by the pesticides. Coming to whether it's species specific or not, so effects of pesticides often vary with the species of animals studied. Not only the level of the intensity may vary, but also the degree also varies. For example, with one may exhibit severe skin sensitivity compared to the other, which may show only mild skin irritation. So it is species specific and as well as it is individual specific also. One animal species may metabolize a pesticide to a non-toxic metabolite, whereas another species may not be able to do so. And coming to individual specific, yes, toxic effects can vary with the size, sex, age, and general health of the test animals. So here we can see the best example in the recent days is COVID. Many of the people are able to tolerate it, but equally many of the people are not able to tolerate it, and it was like very much sensitive to few, but it is like resistant to few. So here itself we can see, maybe it may be in the case of the virus, but here what we wanted to say is that everything is like individual specific, and there the concept of the personalized medicine also arises, indicating that every individual is different. Response of the animal to the chemical. So how this animal responds to the particular chemical. Let us see this. The response of an animal to a pesticide is influenced by a combination of chemical, biochemical, and physiological factors that determines how the pesticide interacts within an animal's body. So the response can range from an immediate effect like acute toxicity to a long-term health issues like chronic toxicity or sublethal effects. So the key factors involved in these interactions include how the pesticide is absorbed, how it is distributed, how it is metabolized, and how it is excreted by the animal, as well as how the pesticide affects the animal's physiological system. So these are all the parameters that need to be considered in order to know the response of the particular chemical in each individual. So we have read like chemical factors that play a crucial role in this. It's the chemical structure. The chemical structure of a pesticide determines its toxicity, yes, the solubility, the persistence in the environment and within an animal. For example, lipophilic pesticides tend to accumulate in fatty tissues, making them more prone to bioaccumulation. And water soluble, that is hydrophilic pesticides, are more likely to be excreted through the kidneys and have a shorter half-life in the animal. So depending on the chemical structure of the pesticides also determines the severity and the stay of the pesticide in the body of the individual. Coming to the chemical stability, some pesticides are more stable and persistent in the animal system. For example, organochlorines like DDT, while others break down more quickly. Example, pyrethroids. So here, uh, DDT, it's a synthetic compound and it has been banned in US like we have discussed yesterday. And pyrethroids are the compounds of the chrysanthemum plant. So definitely natural pesticides are more quickly to break down and get out of the body, whereas synthetic and inorganic things will remain and will be more stable, causing ill effects to the body. The persistence of a pesticide can influence both acute and chronic effects on animals. And coming to the mode of action is also involved in it, like different pesticides target specific biochemical processes in the animal. For instance, a few pesticides like organophosphates and carbamates, uh, they work on neurotoxicity, like inhibiting cholinesterase activity and affecting the nervous system. Whereas some insecticides and fungicides, they work on endocrine disrupting channels and can interfere with hormones signaling pathways. And herbicides may affect the growth and development of animals indirectly by reducing their food sources, which, for example, affecting plants or insects. And coming to the biochemical factors, we have seen the chemical factors and now coming to the biochemical factors. In the biochemical factors, the first thing we have to see is the metabolism, like how this pesticide is getting metabolized after entering into the body. The metabolism, once a pesticide enters into your body, it undergoes a lot of metabolic process and that can detoxify the pesticide or it can activate the pesticides depending on the enzymes and the pathways that it encounters in your body. And several factors influence how the animal metabolizes the pesticides, and some critical factors are cytochrome P450 enzymes, which are very much important in the detoxification of many chemicals, including pesticides. Some animals may have higher or lower activity of these enzymes, influencing how they process and detoxify the pesticides. Coming to the phase 1 oxidation and reduction and phase 2 conjugation reactions, which are seen in liver, they help convert pesticides into more water-soluble compounds for excretion. However, some pesticides are metabolized into more toxic forms as well by bioactivation, which can exacerbate their effects. Glutathione S transferase and other conjugation enzymes help neutralize the harmful effects of pesticides. Coming to the antioxidant system, pesticide exposure can lead to the production of reactive oxygen species, causing oxidative stress. Animals with weaker antioxidant system may be more susceptible to the damage caused by oxidative stress, leading to tissue damage, immune suppression, and chronic diseases. Enzymes such as superoxide dismutase, catalase, glutathione peroxidases play important role in mitigating the oxidative stress because they act as antioxidants to clear this free radical generation, that is reactive oxygen species, which causes oxidative stress. Cholinesterase inhibition, certain pesticides, organophosphates, carbamates, they inhibit acetylcholinesterase, which is an enzyme that breaks down acetylcholine in the nervous system. So this results in the accumulation of acetylcholine, which leads to the overstimulation of nerves, causing symptoms like tremors, paralysis, and death. Here is a picture where we can clearly see how the pesticide is entering into the body through various receptors. And after that, what are the biochemical pathways that it interacts with? Once it crosses the extracellular membrane, so what happens here is, like we have talked, the mitochondrial function will be dysfunction, because here we have seen the cytochrome P40 enzymes are involved, glutenase transferase is involved. So here oxidative stress is generated, mitochondrial dysfunction has happened, inflammatory signals are being popped up, endoplasmic reticulum stress can be seen due to Orvois, the reactive oxygen species, which further leads to the DNA damage and causing cancer, because all the reactive oxygen species that leads to the cell abnormality of the cells, making them produce more number of cells, which in turn leads to cell proliferation, which leads to cancer, converting the proto-oncogenes to oncogenes. Lipid and protein oxidation, oxidative stress. So these are all the things that we can see in the biochemical level what the pesticide is doing, right? Coming to the physiological factors, absorption and distribution. The route of exposure, pesticides can be absorbed into animals' body through the skin, lungs, or digestive tract. Means when we come in contact with the pesticide, or during inhalation, when we take the air through our nose during inhalation, or else when we take through our food, if we eat the contaminated food or water, through that also pesticides will get into the body, that is through digestive system. So absorption rate depends on the pesticide's chemical properties, whether it is a fat or water soluble. Species difference. So different animal species have varying rates of absorption and distribution due to difference in skin permeability, lung capacity, digestive enzymes, and body composition. Age and size of the animal. Young animals, including fetuses and juveniles, may have less developed detoxification systems, and they may absorb and metabolize pesticides differently than adults. Smaller animals generally have a higher surface area to body mass ratio, which may make them more vulnerable to pesticide absorption. After absorption and metabolism comes the elimination, excretion, where kidneys and liver play the major role. The kidneys are responsible for filtering out many pesticides through urine, while the liver processes them for excretion via bile. Some pesticides, especially the ones which are fat soluble, that are lipophilic, may accumulate in fat tissues, resulting in slow excretion and prolonged exposure. Means fat soluble ones are very much toxic than the water soluble ones, because they don't easily get or come out of the body. Bioaccumulation. Animals exposed to persistent pesticides may accumulate the chemical over time in their body fat or other tissues. This is particularly concerning for top predators like apex predators, where pesticide levels can increase as they consume contaminated prey. Toxicological effects on organ systems. So what are the toxicological effects on organ system? There are different organ systems that we have: nervous system, endocrine system, immune system, digestive system, and also how these pesticides are going to affect these particular organs we are going to check here. Nervous system. Many pesticides target the nervous system, especially organophosphates and carbamates. For example, they inhibit cholinesterase, leading to overstimulation of the nervous system. This can result in tremors, seizures, paralysis, and death. Endocrine disruption. Some pesticides can interfere with the endocrine system by mimicking or blocking the action of hormones. This can lead to reproductive issues, altered behavior and developmental problems. For example, some insecticides may affect estrogen or androgen signaling pathways in animals. Immune system suppression. Certain pesticides, especially in chronic exposure scenario, can suppress the immune system, making animals more susceptible to infections and disease. Liver and kidney damage. Liver plays a key role in detoxifying chemicals, but some pesticides can damage liver cells, impairing their function. Kidneys are also affected in some cases, leading to nephrotoxicity and kidney failure. So these are all the different systems where organ systems where pesticides act and leads to ill effects. Coming to genetic and evolutionary factors, species-specific responses. So different species may have different sensitivities to pesticides due to genetic variations in enzyme activity or physiological traits. Some species may have evolved resistance to certain pesticides through natural selection, while others may be more vulnerable. So that is what species specific is. Like some species, that particular pesticide of that particular concentration may not be lethal. If the same pesticide with a similar concentration, which is not lethal to one species, if it is given to the other species, that particular species may have vulnerable effects, which may lead to death. So we cannot say that one particular drug with that particular concentration will be safe for all the species, because here there is like lots of difference in that enzyme activity, physiological traits, depending on the nature of the selection, natural selection. So there are like many factors that play the role. So it is species specific. Acquired resistance. So coming to acquired resistance, pests like insects and rodents, they can develop resistance to pesticides over the period of time through genetic mutations that allow them to survive exposures that would normally be lethal. So that is acquired resistance. If you keep on giving the pesticide of that particular dose to the insects and rats or mice, upon the time they become resistant to the drug, so that that drug will no more be lethal to that particular animals. It will not show any significant effect that we want to see in insects or rodents. That is what the resistance or the acquired resistance mean here. And resistance mechanism can include changes in metabolic enzymes, altered target sites, or ability to detoxify the pesticide more effectively. Coming to behavioral factors, feeding behavior. If an animal's usual food source is contaminated with pesticides, it can lead to direct ingestion of harmful chemicals. Whereas some animals may alter their feeding behaviors to avoid contaminated food, but this may not always be effective. Habitat use. Animals may change their habitats or migration patterns in response to pesticide exposure. For instance, birds may avoid areas where pesticide application is frequent, but this can reduce their available habitat and food sources. The interaction between animals and pesticides is a complex process influenced by a combination of chemical, biochemical, and physiological factors. Factors like chemical structure, metabolic enzymes, organ systems are involved, like species-specific, age, genetic makeup, all are the crucial roles that determine the animal's susceptibility to pesticide exposure. Coming to the chemical properties of the pesticide, every pesticide and each pesticide is unique. Pesticide molecules differ in their chemical structure, size, shape, stability, and electronic charge. This difference determines how pesticide function in the human body. So each pesticide is different. Even though they may have the similar structure, their function might be different. For instance, the chemical structure of some pesticides make them fat soluble, while those of the other make them water soluble. The significance is that the former would be stored for various lengths of time in fatty tissues, while the water soluble might be rapidly eliminated through urination. The shape and electronic charge of some pesticide molecules allow binding to critical site receptors in cells or on cell membrane. These and other variables often lead to significant biological response to pesticides. Coming to the absorption into the body, the pesticides, when they can enter the human body through several routes, including inhalation, ingestion, dermal absorption, eye